What type of sugar is in DNA? And in RNA? And what shape? And in function?
Nucleus. Storage. Oh, I did it wrong.
The types.
No, 8. 1. 3. Oh, that's kind.
And what bases can we find in
Nucleus.
DNA?
Is it AU or AB? Oh, it's AU.
What was it you stand for?
Uracil.
Is it spelled as it sounded?
URACIL. And what we didn't fit on here is location. Where's our DNA located? And where is RNA located? Excellent. So I do believe we stopped roughly there yesterday.
Yeah.
And I told you there were 3 different kinds of RNA, but I told you what their names were, but we didn't look at their shapes. The 3 types of RNA are absolutely essential to the process of making the final product, of making DNA useful. Their shape dictates their function. So you'll know what their function is based on how they're shaped. It's important. It matters. So mRNA is the one that we say is a single strand. So you'll look at it here. This is mRNA. It is a single strand with a sugar phosphate backbone. But instead of a deoxyribose and phosphate backbone, it's a ribose and phosphate backbone of single unpaired nitrogenous bases. So that's mRNA. rRNA is actually the main component in ribosomes. Ribosomes are just made of 2 stretches of RNA. One we call the large ribosomal subunit. It's the big half. And one we call the small ribosomal subunit. That's the small half. And that's all ribosomes are made out of. They're produced in the nucleolus and then sent out into the cytoplasm where it attaches to ER. So if you remember seeing ER in our cell theory chapter, it was that stuff that looked like a ribbon that was outside of the nucleus, and we labeled some of it rough ER. That's what makes rough ER rough, is that RNA has attached to the outside. For some reason, and I never really understood this, most biology textbooks say that tRNA is shaped like a key. I must not use the same kinds of keys that any of them use, because to me that looks like a T, like the letter T up here. I don't know that I get much of a key out of it, other than the fact that down at the bottom it's going to fit like a lock and key when it gets used, which could be their reasoning, but I've never looked at that and been like, oh yeah, that's definitely key-shaped. But that's what they say for transfer RNA. So those are your 3 types of RNA. Each one of these has a different role in the process of making a protein. So yesterday you told me, or we talked about how DNA is the storage, RNA is the temporary thing that gets made, and protein is the final product. And you remember way back from chapter 2, this would be roughly like October. It's totally fresh in your mind, right? October? October? All right. Let's try this. You had a chapter called biochemistry,
Oh, yeah.
and you learned 4 major biomolecules: carbohydrates, lipids, nucleic acids, and proteins.
I remember now.
And you should hopefully remember that all of those big biomolecules are made out of tiny pieces, right? Nucleic acids, their tiny pieces are called nucleotides. RNA is still a nucleic acid. So what's its tiny piece? What's its monomer? Still a nucleotide. Here's going to be the one that might be a challenge.
Nucleotide.
No.
Protein
Acid.
is
Nucleic acid.
amino acids.
Dang it. She was trying to trick us.
So somewhere in this process of going, and mind you, I do want to just say, this is a bit oversimplified for how it actually works, but for the purposes of understanding, it's okay. Somewhere in this process, we have to rewrite it and then switch languages. Language 1, language 2. So this arrow right here, that's a process that we call transcription. Remember yesterday we said to scribe means to write. So we rewrite it. And then at this second arrow is the biological process that we call translation. The process of transcription occurs in the nucleus. And I know it occurs in the nucleus because it uses DNA. And that's where the DNA is.
Is that recording your voice too?
Yes.
You're putting on plus forward?
Yes.
So if we just like say something random, you could hear it in plus forward.
Yes, so do your parents. That has been very helpful at parent-teacher conferences in the past.
So. No, that's right. Yes.
From the nucleus, the process of transcription is going to take DNA, turn it into the specific kind of RNA that sends messenger RNA. That's going to be taken out of the nucleus to the cytoplasm. Yep.
Does it have to be lowercase m?
Yes.
Where do you get that amino acid from?
Out in the cytoplasm, transfer tRNA is going to bring all of the amino acids. So it carries them to it. Mm-hmm. All right. Oops. Here, an enzyme here called RNA polymerase. What was DNA polymerase's function in DNA synthesis? Well, what's its job? All right, so it sticks something to another, specifically base pairs, right? RNA polymerase is going to do something very similar. But let's take a look at it. Some biology art.
We have to draw.
Hopefully by now, we're comfortable with that one.
Double helix.
Excellent. Of what? one gene, so we covered genetics, right? We talked about dominant and recessive, and one gene simply represents one small section of DNA. So this is one single gene. And what's going to have to happen is that the helix is going to open. And after the helix opens, only one side of that gene gets read. We call it the template. So the helix opens at a region we call the promoter. Every gene has a promoter that's not part of the gene itself. It's a section of nucleotides that occur just before. The reason for that being, think of it like those recipes that we talked about. If I didn't make sure I copied the entire recipe and part of the recipe got left off, it makes the rest of the recipe useless, right?
Yeah.
So you include a little bit of the page. Like if I'm taking the picture, I make sure my picture includes more than the recipe. And then later I can crop it off the photo. So that way I know for sure I've gotten the whole recipe. DNA will do the same thing. It's going to open up at a region that is just before the gene itself. And when it's just before the gene itself, we can make sure that it includes the entire recipe. And that's where the T will pair with, C pairs with, G pairs with, A, C, T, A. That's what happens in the process of transcription. RNA polymerase, the enzyme, is simply going to make a strand. Once that strand is made, it's released from the complex. Then the DNA can make a helix again, so it seals back up. And this strand is sent, actually it's edited first, and after it's edited, it's sent out of the nucleus to a ribosome. Fairly simple, right? This is the easy part. The only other thing that's going to happen is a little bit of editing. There are things or regions within every gene that get edited out. You guys have written papers before, right? And when you write papers, does your final draft always include everything you've written? You edit stuff out, right? And there's multiple ways you could have edited that thing. Now, what's interesting about our genes is there's multiple ways genes can be edited for different uses. That's why I said that whole like DNA, RNA, protein thing is a little more complicated. There's multiple versions of editing. But here's kind of how it works. In the editing process, before it leaves the nucleus, I do believe one of you asked me what is splicing. Here it is. What's it mean to splice something? You guys don't ever have to do it anymore. Cut it up, right? Splicing is the term that they used to have to use when you edited video, like way back in the day when video was actually like on like film. They used to have to cut it and splice it together. So the process of splicing is taking things, cutting them, and then re-adhering them in a different order. It's editing. So in the process of transcription, a cap gets added to the end of the RNA. And sections of the RNA get edited out. So they get spliced out. You'll notice here I grouped my RNA in threes, right? It's very deliberate. It's because when we read in threes. They're called codons. We read 3 base pairs at a time. We can label the sections that we keep of RNA, we call them exons, and introns are the part that get edited out. So the splicing mechanism takes any introns and cuts them, or takes any exons, keeps them, and takes any introns and cuts them out. So after the cap is added, I would have keep my exons, and then we put a tail on the end. The function of the cap and tail is so that when the mRNA makes its journey from the nucleus out into the cytoplasm, it doesn't get damaged in any way. It's like putting something in an envelope. It protects it so the message arrives to its destination complete and intact. That's RNA editing that makes mRNA. So we've spliced out our introns and kept our exons. I've repeated that a few times because you'll notice that's the opposite of what you want it to be. EX in your mind usually means exit, right? When you think of EX, you think of things that are leaving, are going. And when you think of in, you think of things that stay, that are included, that are kept. Here it's the opposite. Introns are what leaves, and exons are what's kept. So I always just remembered it by thinking about it being opposites. Is when we're going to switch languages. So we're going to go from that mRNA nucleotide language. We go from there to the amino acid language. And we call it protein synthesis because that's overall what's going to happen here is proteins are going to be made. This process occurs attached to ribosomes. So remember how I said that all of our forms of RNA have a really important job here? Well, mRNA carries the message. Ribosomes, ribosomal RNA, gives us a place for the reaction to occur. We translate it using a thing called the genetic code. Now, you understand codes because I'm sure, I don't know, it's probably like for me, 6th grade history, world history-ish, except they called it social studies at the time. Then you talked about hieroglyphics, right?
Oh yeah, that's awesome.
And all of
The
you probably
Egyptians.
had to take your name and translate it into hieroglyphics and made like a bookmark out of it, right? Some things never change. Like, well, I guess okay. This is Egyptian.
That was so
What else?
In 7th grade, all I remember studying is Egypt, Canada, and Mexico. Our teacher only focused on stuff that she liked.
science.
This is where the now where did the 6 months?
The teacher that was what?
You're saying there's 4 countries in that.
I don't
So,
know, she just said Canada.
seems like an odd thing to be like, yes, Canada. Again, I like kindergarten. So, it's much like the idea of using a, even just one of, I don't know, I feel like when you're a little kid, like the back of the cereal box always has like a secret code cracker.
I still do it.
Those
So,
are hard.
you understand that there's like a code that if you take one thing and you use this tool to help you translate it, you can turn it into a different language. We do the same thing here. And we call it the genetic code. And it looks like this. So this is the genetic code. I said we look at messenger RNA 3 bases at a time, and those 3 bases are called codons. So what we would do is take a strand of messenger RNA, let's use this one, and we would look at the 3 bases, A, U, G. We take that to the genetic code, The first nucleotide was A. The second nucleotide was U. Follow it to the box that corresponds. AUG corresponds to the amino acid we call methionine. But we're not going to write methionine when we write down the amino acid code. We would just write the first three letters, MET. So there's the first one, methionine. AUG is also the codon that says start. There's one start codon for every protein, AUG, and there are 3 stop codons. Find the stop codons.
What's a codon?
A 3-letter section of mRNA. All right, there's one. Stop.
Do you want us to write this down?
UAG and UGA. On your test for protein synthesis, I will give you a genetic code. So you're either going to get this picture, which is my preferred picture, the square picture, or I think your textbook, I'm pretty sure, has like this crazy thing that looks like a sun. Pretty sure, let's check. Or, this is the one your textbook has, where you start at the center and find the first letter, A, then you look for the second letter, U, and then the third, G, and trace it down to methionine on the side. Any preference as to sun? Take a look at 303 for the sun, or here for the table. I'll let you vote which one gets put on the test, because I don't care. Table, sun.
Table.
Both does seem like a little bit of a waste of paper. Table?
Table.
I think the table is easier to read myself because it has it like right in front of you, so even if you're not able to follow the first nucleotide, second nucleotide thing, you can at least read the whole table for the correct combination. It'll take you longer, but it's possible. Let's take a look at the second one, CGU. Find that here.
CGU. C. Oh, R. R.
So this is arginine. How many different amino acids are there?
At least 7.
R.
Well,
16.
phenylalanine, leucine, serine, tyrosine, cysteine, tryptophan.
Oh.
Yes, the stuff that makes you sleepy in turkey. That's the stuff. Arginine. So now we're on 7, right? Glutamine, histidine, proline, isoleucine, methionine, threonine, asparagine. We have not counted lysine yet. We did not count, oh, we did count serine already. We already counted arginine. Glycine, glutamic acid, aspartic acid, alanine, valine. There are 20 amino acids, but way more codons than that, right?
Yeah.
There's more than 20 codons. In fact, there should be 64 on this table.
Whoa.
Whoa, whoa.
That's why we say the genetic code is a duplicate code or a repetitious code. And it's actually really convenient for us because if in the process of transcription a mistake happens, say RNA polymerase puts the wrong base pair in that order. If the mistake that it makes is one where the codon goes from GGU to GGC, the final product still has the correct amino acid. So you're not sick. So there's not a problem with that particular organism and the protein that it makes. That's really, really important for survival, that it is a duplicate code. Read the next one for me. GGU, where is it?
G.
The line.
Lysine. AUA?
Wait, you look for the first 2 letters?
Yeah, like which one
So-
goes first?
First nucleotide, A. Second nucleotide, U. Third nucleotide, A. AUA is isoleucine. Now remember, I don't expect you to memorize the table. You will be given the table. It will have the 3-letter abbreviation on it. So the corresponding answer to this would look something like this. We said methionine, then after methionine, you told me what? Arginine. And then you just told me isoleucine, right? No, this was isoleucine. This was glycine. So that's what your answer would look like. You do not need to waste all of the time writing the whole word out. These are the accepted abbreviations of every amino acid. Any scientist who looks at it is going to know which amino acid that corresponds to, so don't waste your time. This matching is done by the third player of RNA we haven't mentioned yet. mRNA carries the message. rRNA provides us a place. tRNA is the one that brings the matching amino acid to the codon. So tRNA brings the matching amino acid to the codon.